first up today for our research presentations, we have Stephan Frangakis, and he's going to be speaking on the challenges and advantages of trans-ethnic genome-wide association studies in diverse cohorts. Thank you.
Hello, everyone. So a little about me. I'm an assistant professor in the Department of Anesthesiology, so I'm an anesthesiologist. I also have can you guys hear me? I also have a PhD in genetics, so my interest is in the merging of genetic data with things that are important to me as an anesthesiologist, namely pain, post-surgical pain. So for pain studies, it's historically been pretty hard to get a good large cohort, because in order to determine what someone's pain is, you have to go and ask them, and this usually requires prospectively collected data or surveys. Go ask somebody what their pain is. We have many types of pain scales that have been validated, but you still have to do that face-to-face, asking somebody what their pain is. And so what's great about what we have here at MGI is we have this huge genetic cohort. We also have a small subsample within the MGI called the analgesic outcome study. And so this comprised of about 3,000 University of Michigan surgical patients. So they're coming from surgeries that are, there's abdominal, gynecological, orthopedic makes up the vast majority of it, and then breast and thoracic surgeries. All these patients have prospectively collected survey data, so they get surveys at baseline, so right before they're going in to surgery, and then we follow them up with survey data at 1 month, 3 months, 6 months. We also have a small group that has surveys at 2 weeks too. And then just like every other MGI patient, their blood is collected in the preoperative area and it's biobanked. It's then imputed through the MGI system. So this is just a rundown of our AOS cohort. Slightly majority female. You can see that there is majority older population over 65. This is because a large percentage of our patient cohort is coming from orthopedic surgery. So usually older when you have a total hip or total knee replacement. So what's very pertinent to this talk is you'll see in our cohort we have greater than 90% is white, Caucasian, or European ancestry, and about 5% is Black or African American. So this is self-reported race data. And we also look at what their baseline pain is and say a good proportion of these people already have pain at baseline. Again, probably because a lot of them are coming from orthopedic surgery, so you're not going to get your knee replaced unless it's already hurting you. And so there's a quick graph just showing what's so special about our cohort is that we have data from when they first come into surgery, and we have data from 3 months later. Most people are not able to get this linear follow-up data. And you can see most patients that have pain, they drop down to 0 or much lower. There's a small percentage of patients that may start from 0 and then have pain after surgery, or continue to have pain throughout their post-surgical process. And these are the patients that we want to get. So we initially took their pain scores. We did a little bit of linear regression to try to normalize out their pain scores, and then we ran a GWAS, and we got this nice GWAS that has a lot of peaks to it, and we were very excited and very happy that we potentially found some genetic factors associated with post-surgical pain. For those of you who are biostatisticians or have seen a GWAS before, might be looking at this and saying, something looks a little funky, which is what our biostatisticians said, kind of raining on our parade. We looked at the QQ plot. For those of you who are biostatisticians, we'll look at this and say, yeah, something's kind of funky. So that was a little upsetting, but I'm glad that we saw it. So something was a little weird with this data. Like, okay, how can we figure out what's going on? We then went and did some permutations, and basically what permutations of the data is when you take the phenotype, you scramble it, so now the phenotype scores and the genetic identifiers are totally mixed up, and if there's something inherently wrong with the data, maybe you might see peaks. And so we did our permutations and everything was flat, kind of what you would expect if the data was good and clean. You mix everything up, you don't see a signal anymore. It didn't really assuage any of our concerns. So then we said, okay, we're supposed to be controlling for race, but let's just do an experiment. We took our cohort, we separated out by self-reported race, and so we looked, this is just those who self-reported as white or European. and now we get no real signal. We had a smaller cohort that was identifying as African American or Black. Again, no real signal. So something's going on when you look at just white patients or just Black patients. There's no signal. When you combine them, something's popping up. We thought, okay, we did these permutations. And so initially what we did is we did a whole group permutation. And see if I can. So whole group permutation, we have our white cohort, we have our Black cohort, we mix everything up, and so everything's mixed up together. So that's what we did initially when we saw, no signal to our permutations. We thought, what if we take the white cohort, permute it just the white people, and then take our Black cohort, permute just those self-identifying as Black or African American. Still keep the racial cohorts separate, but have permutations. And we did that, we found those signals. There shouldn't be signals if you mix up all the genetic and phenotypic data, but there was because we kept the racial cohorts separated. So this kind of tells us that there was something wrong with our data, or something that was affecting our data that was based on racial cohorts. So this is our original GWAS. We said, okay, let's investigate this a little bit more. Here are these signals that we saw, and we were initially very excited that we found some genes associated with pain. We went and looked what's actually the allele frequency between the racial cohorts of these sites. And you can see here the allele frequencies in some of these high, the signals that we saw is about 10,000 fold difference in the allele frequency in, actually about 1,000-fold difference in the allele frequency between the Black identifying patients and those self-reporting as white. So basically what we were finding were alleles that were segregating with self-reported race. So just to kind of delve in a little bit, so there's a lot of statistics, I'll just kind of give you the broad overview. I had tried to correct for kind of the self-reported pain scores that people were doing by saying, okay, we're looking at the difference in pain score from month 3 to the baseline. So what's their pain change from baseline to month 3? And so if someone, is reporting, if someone doesn't report pain that much, we'll at least self-correct them because we're kind of getting the difference between them versus somebody who is reporting pain higher than others. We're taking that difference, so that should self-correct. And you can see the difference is actually pretty similar between the two racial cohorts. The problem is that I corrected for baseline and month 3 pain scores. You see those are inherently different. And what that led was a different residual value when we did our linear regression. And that led to, when we normalized it, a different normalized value just through the overall cohorts on average. So that's basically what I was doing, was I was specifically isolating variants that associated with self-reported race instead of pain. So some of the problems with this, with the analysis that I carried out was that Black or African American subjects made up a small subset of the overall cohort. So they have overall a different genetic background at certain sites, as we saw from our allele frequencies, as well as slightly different phenotypic expressions. So when you do your linear regression, the computer's like, oh, this is a small 5% group of patients. We can just kind of throw them out. We don't have to, it doesn't change the math of the linear regression, whether it is concerned with this cohort or not. But then when you plug that into our GWAS, it has a notable effect in our GWAS results. So what do we do here? We have, and you guys saw from the now free 7, we're going to have an even greater percentage of a diverse population. So historically, genome-wide association studies have been mostly European ancestry-centric. So this looks at, see, the total number of studies. More than half of the total number of studies are European cohorts. And then when you actually spread that out into how many people are in each study, you can see it's more than 3/4 of patients in genetic studies are European ancestry, with a small amount of Asians and then even smaller amount of Black or African American or African. So what happens when you have a multi-ethnic cohort, you want to combine the genetic data from these different ancestral groups. And so the goal here is to identify findings that are applicable across genetic ancestry to everybody. So if I find something that's only applicable to Caucasian patients or only applicable to Black or African American patients, that's much less useful than something that's applicable to all humans. So there's some drawbacks to these multi-ethnic cohorts, as I just showed what happened. So there's heterogeneity across populations. And so this can introduce confounding variables, or it can be termed genetic noise. So we can try to correct it, but sometimes if the ancestry has a separate association with the trait, as opposed to ancestry going to a SNP going to the trait, then you can get difficulties in correction, as what we saw. So when you stratify your populations based on, this is a white Caucasian subcohort, this is our Black African American subcohort, this is our Asian subcohort, this stratification itself can result in false positive or negative, as you saw what happened with me. And then because of this different genetic background, there's differences in linkage disequilibrium structure, and this can affect your interpretation of results. So here's an example of what we hope to find is that you have all these different types of SNPs from different racial cohorts, and they all affect one variant. But what really is probably occurring is that because of the different genetic backgrounds, the different SNPs and different variants affect different sites differently. And then there's also the problem of what's the definition of race? How do you determine if somebody is Black or white or Asian based on what they say? Some people can be more than one race, surprisingly. You can be Asian and you can be Indian too, as we have heard recently. So some other drawbacks. I'll talk about the benefits soon. So there's some things we have to work through. Statistical methods so far, because remember, 75% of genetic population has been white or European. They're designed for homogeneous populations, so they don't work well for diverse data sets that have a lot of background noise. Then, of course, there's difficulty ensuring that what you're finding is applicable across these diverse populations. And of course, just all of our reference panels that we use to determine what's a variant, what's a SNP, this is made in specific populations and cohorts. So now the advantages of having a multi-ethnic cohort. Why do we want to recruit a more diverse population? Well, we kind of want to expand our results past just the historically European ancestry. So here's an example which it's actually really helpful. So this is a polygenic risk score. So this is when you actually look at the polygenic risk score for these two populations, you have Latino and then non-Latino white. You can see that there's a different spread of polygenic risk score in this example. So if you are able to identify the different racial or ancestral cohorts, you can get more specific results for each cohort and something that's actually more applicable. If you're able to find something that's applicable to everybody, then everybody can benefit from it. And then you also want to, it'd be interesting to find traits, genetic variants that are influenced differently depending on ancestral background. And so if you do find something that is applicable across all humans, you know that what you found has more statistical power, is more effective. So this is an example of something where a signal from the European cohort is very broad. When you combine in a signal from the Asian cohort, you can identify the site across these SNP markers, the site that is probably more effective. So by adding more diverse population to your cohort, you can get more specific results. That is, if you know how to handle it. And because you're adding more cohorts together, you're able to find things that you weren't able to find before. Let me just show. All right. again, like we said, so the whole point of what we're doing here is for precision medicine. You want to be able to find things that you can apply to the patient in front of you. All right. So ways that we can do this, and something that we are working on now, is what's called a meta-analysis, trans-ethnic meta-analysis. So what this does basically is combines GWAS data between different populations. So what you're aiming to do is to find variants that are shared between these two populations. Right. You can combine it in a fixed or random effect model, but there are some limitations to it. So again, like we said, there's different linkage disequilibriums across the population, so it's kind of hard to combine these two results sometimes. It also kind of underestimates the effects of the specific populations. So while you can find individual variants that might be effective in each population, combining them together in different ancestral backgrounds can be complicated. So you can use what's called trans-ethnic meta-regression. So it's just an extension of meta-analysis. So it also models the variation in allelic effect with different ancestral backgrounds. So it incorporates the population structure. So you can find specific alleles that are shared as well as alleles that are variant depending on this ancestral background. So because of that, it has lots of advantages over meta-analysis. It's a little bit more statistically intense and computationally involved, but it is effective. So just to sum up, the cohort that we have with MGI and what we're trying to build even further has some challenges with it. But the benefits of including a multi-ethnic, multi-cohort these outweigh some of the drawbacks to it. But you need to have proper methods to mitigate these challenges. And the results that you do end up finding will be more impactful. So first, we're working on improving our statistical methods, getting greater, to be able to apply the methods that we have really developed for European populations, to be able to apply it across all human populations, expand our reference panels, and we need to encourage the diversity in our research in recruiting as well as the people conducting the research. So a lot of people I need to thank. Jad Brahman, my PI, has been really helped drive this project forward. Matt has been one of the biostatisticians with the School of Public Health that I've worked extensively with, who initially looked at those GWAS results and said that something was probably wrong, we should look into it. And really, MGI, Precision Health, and the whole department have been very helpful for us. Thank you.